So within predictive microbiology, we want to predict how a microorganism behaves in a certain environment depending on the processing or the storage period of a food. Now, to develop a model, it takes a lot of time, and within the course we go through the different items that you need. So first, it is important to have a real good experimental design to know what will be the aim of your model that you develop in the end. Based on that, you make your design, and after that you go to the data generation. The data generation of a predictive model takes a very long time. You have to do a lot of experiments, a lot of replicates in very standardized conditions to be able to find a trend and to be able to model it then afterwards. The modeling itself is applying some mathematical equations, differential equations and other equations to the data set that you obtained in your second step. Now, those equations, they are taught in theory during this course, but as well they are practiced by computer exercises, so that you know how to do it after following this course. In the last step, you do that. There is a validation of your model by doing some extra tests in a food product, for example. Also, data concerning this are given in the course, and you can check whether the developed models are correct or not. So, in general, after following the course, you should be able at least to define yourself and to develop a new model and to use it then afterwards in practice. Within predictive microbiology, we try to predict the behavior of microorganisms in a food for a certain process that the food undergoes during processing or during storage afterwards. And we distinguish two larger types of major types of models. It are the kinetic models and the probabilistic models. Kinetic models, they really describe the growth rate as a function of time. So the evolution in your population density as a function of time, while a probabilistic model rather predicts a chance that a certain event will appear or not appear. For example, the microorganism will be able to grow or will not be able to grow. In this course, we give those mathematical equations as well as the practical exercises, so that immediately you can apply all the knowledge that you have in the environment, which is important for the food industry or for the regulators, for the government. But we have to say that predictive microbiology is no magic. So it's very important that all the predictions that you make, that you can also judge them very critical, whether they are correct or not. And a lot of attention has to be paid to that as well, to critically see what comes out of a prediction. So to develop models, it takes years to make them, but to use the models, it only takes 1 minute. But also then it is important that you have good knowledge on interpreting them, and therefore a lot of industries come to the research centers to evaluate the models and to give not only the outcome of a model prediction, but also the interpretation. And you have a lot of freeware software packages, but as mentioned before, they are no magic. So also there you need some knowledge about food microbiology in general in order to interpret them in a correct way.